Let's now focus our understanding of excitation and inhibition by working through a couple of very specific examples. First, let's consider the classic example of a fast and strong EPSP, the action of acetylcholine on nicotinic acetylcholine receptors at the neuromuscular junction. As you might guess from their name, nicotinic acetylcholine receptors can be opened not only by their endogenous neurotransmitter acetylcholine, but also by the exogenous compound nicotine, as found, for example, in tobacco products. Independent of their origin, both nicotine and acetylcholine act as stimulants at these receptors. At the neuromuscular junction, this postsynaptic stimulation results in an above-threshold depolarization and muscle contraction. During this process, about 50 to 90 acetylcholine-containing vesicles, or quanta, are being released into the synaptic terminal of a single motor neuron innervating the neuromuscular junction. The released neurotransmitters act with high chemical specificity on the postsynaptic acetylcholine receptors, which are perfectly aligned with the nerve terminal. This high degree of spatial specificity is crucial to the success of a strong and fast excitatory synapse, for reasons we'll understand shortly. So how does the action of acetylcholine on acetylcholine receptors lead to an excitatory potential in the postsynaptic cell? To understand how the EPSP is generated, let's take a detailed look at the structure and function of the nicotinic acetylcholine receptor. The acetylcholine receptor is a ligand-gated ion channel. That means that it opens when a specific chemical, called the ligand, binds to it. Like many channels, it's composed of a constellation of separate proteins termed subunits. It consists of 2 copies of the alpha subunit, as well as 1 copy each of the beta, gamma, and delta subunits. These subunits are assembled in the cell membrane in such a way that the overall protein complex creates a central pore that's about 0.8 nanometers wide, enough space to let small ions and their water entourage or solvation shell pass through. If looked at top-down by a technique called electron microscopy, which we encountered previously in this lesson, the fully assembled acetylcholine receptor resembles a donut, with the central pore being clearly visible. If looked at from this side, the acetylcholine receptor shows a large extracellular membrane-spanning domain, as well as a smaller intracellular domain. Each fully assembled acetylcholine receptor has 2 binding sites for acetylcholine neurotransmitter molecules on the alpha subunits on the large extracellular domain. While the entire acetylcholine receptor is composed of 5 subunits, each individual subunit has 4 membrane-spanning domains, termed M1 through M4. It's important to note that the subunits are arranged in such a way that each M2 subunit lies in the central pore. So how does this channel function? As we already mentioned, the acetylcholine receptor is a ligand-gated ion channel. It's named such because the binding of a chemical ligand, acetylcholine, to the binding sites on the 2 alpha subunits on the acetylcholine receptor causes the ion channel to open. This ligand-induced change in the 3-dimensional structure of the receptor, termed a conformational change, allows the flow of ions through the central pore of the channel. But this happens only when both binding sites for acetylcholine on the receptor are filled. If none or only one of the binding sites are filled, the receptor won't change its 3-dimensional structure. No central pore will open, and the ions can't pass through. How does all this molecular action relate back to the generation of a fast, strong EPSP at the neuromuscular junction? As we already hinted at the beginning, a single EPSP doesn't equate to a go signal for an entire neuron. Generally, many EPSPs have to sum up to generate an action potential in a postsynaptic neuron, or in this case, a muscle fiber at the neuromuscular junction. In the case of the acetylcholine receptor, the excitatory postsynaptic potential is the sum of currents generated through many acetylcholine receptor molecules. For comparison, the current generated by a single acetylcholine receptor equates to approximately 2 picoamps, but a typical synaptic current at the neuromuscular junction equates to approximately 600,000 picoamps. This means that roughly 300,000 acetylcholine receptors are active at the neuromuscular junction during synaptic transmission to generate the observed strong EPSP that can trigger an above-threshold response. Interestingly, while these channels open nearly at the same time due to a near-synchronous release of acetylcholine at the specific synapse, each individual channel closes randomly. During the rising phase of the EPSP, the postsynaptic neuron is depolarized to about negative 35 millivolts. This change in postsynaptic membrane potential is due to the opening of many ligand-gated acetylcholine receptors, generating a strong synaptic current. Cumulatively, this current is strong enough to reach threshold and trigger an action potential through sodium influx. This sodium influx is mediated by voltage-gated sodium channels at the axon initial segment of the postsynaptic neuron. During the falling phase of the EPSP, which brings the membrane potential back to rest at around negative 70 millivolts, the acetylcholine receptors close randomly in time. In addition, potassium-conducting leak channels, which are always open and present in the postsynaptic cell membrane, aid to repolarize the membrane potential back to rest, readying the postsynaptic cell for another cycle of neurotransmission.